Okay, so thank you very much for having me here. Today I just want to walk around some of the new material technologies that we have been working on at Imperial College, but more broadly sort of within the field of bioelectronics. In particular to point out some of the areas that we kind of struggle with that I think, you know, through interactions with Royce and through industry, we have an opportunity here to actually really change and make it like a big step change in the field that hasn't actually moved very much in the sort of the past 70 years, to be honest. So when I talk about polymer bioelectronics, I'm talking about all these sorts of devices. I'm talking about implantables and wearables and the way in which they electrically interface with the body. And one of the things that's become more and more interesting over the sort of the past couple of decades is learning that most cells have some sort of intrinsic electrical activity and can be influenced. Their cellular mechanisms, their ionic channels can be influenced by electricity. What's critical about that is that when we actually interact with the body, when we start to try and record signals or push signals in and change what those cells are doing, we have to do so in a way that's informed by the biology. And historically, it really hasn't been informed by biology. It has been informed by necessity, by things like the pacemakers, which were developed out of sort of wartime and a need to restart tissues that were electroactive, and we use very rudimentary metal technologies. Sadly, that actually put us in a position where then for the next 50 years or more, we get made more and more devices based on that exact same technology without looking at what was actually happening in the biology, informing it with the biological milieu. So what we look to do, at least sort of within my group and within the field of polymer bioelectronics more broadly, is to develop these flexible solutions, soft solutions, things that are informed by the way in which the cells actually communicate to make a better interface for medical devices. Conflict of interest is I do have a startup in this space, predominantly actually in the wearable space, and there's a really good reason for that I'm going to touch on, I think, as I get towards the end in the commercialization side of things and why we have quite a few hurdles in our way to getting some of these really revolutionary technologies working in medical devices. So clinically, I mentioned earlier the pacemaker is something that came out of war times and eventually was commercialised and became general use in the 70s. And effectively, this used platinum electrodes. So at the end of those two leads is two platinum electrodes, and then that's just a pulse generator unit which sits far away from your target tissue. All of the devices we have here that have come about, the cochlear implant came next. It was commercialised, sort of developed in the late 1970s, commercialised in the late 1980s. Again, platinum electrodes. This device in particular was developed by Cochlear Limited in the 80s with 22 platinum electrodes. And that was used to render all the different sounds that we have and language, and then that has enabled people to, have the return of sensation of hearing. It's enabled people to learn to speak, so it's done some amazing things. But today, guess how many electrodes we have in a cochlear implant? 22, in the exact same amount. And they're still platinum because we haven't made that step change to change the way in which we interact with the body. Instead, we've reimagined this device in different ways. This is a deep brain stimulator. It is a straight cochlear implant jammed down in the deep brain with the same pacemaker unit stuck on underneath the skin. And we then reinvented it for a peripheral nerve. So you now have these things go down in the peripheral parts of the body. Recently, we did something minorly more exciting, and this is a leadless pacemaker. So we've actually found a way through, this has been off the back of the acceleration of the microelectronics industry. By miniaturising chips and being able to get that very small, we've actually been able to solve some of the issues with pacemakers by making them very, very tiny and having them indwelling in the heart. They don't work for all patients, and the less exciting thing is this is off the back of electronics advances and hasn't taken any intuition from the material advances that we've had in the time being. So it is still platinum electrodes stimulating just without the leads on a very, very small, tiny, tiny pacemaking unit. But that doesn't mean we haven't been doing these developments. The developments have been driven in the biomaterials and the bionics space to try and make new exciting devices that platinum can't deliver on. So one of them is the bionic eye, and way back in my PhD days, I worked with the bionic eye team in Australia who developed this particular device here. And the challenge was we needed to get many, many, many more electrodes onto a device. So if you want to put useful sight into a patient, 22 electrodes isn't going to do it because that means they're seeing with 22 pixels effectively. And if you think of a computer screen with 22 pixels, you ain't doing anything with that, are you? It's very, very rudimentary. So they started off with ones that were sort of 60, 100 electrodes, but realistically to get to useful vision, when you go and speak to patients, what they want to see people's faces. They want to see their children smile. They need thousands of electrodes, and platinum can't do that. I'm going to tell you why in a second. They're just going to put a few of the other devices here. This is called a UTAR array. This one came off, again, around silicon chip industries. They learned how to etch and make very nice three-dimensional structures. And the UTAR array is something that's been used very good for recording in research. And they're starting to figure out how they can use it in humans, but it's still a bed of nails. It's a very stiff, hard silicon device, which basically doesn't take into account the fact that the brain is a very, very squishy, moving piece of tissue. This is a stent trode made by the Synchron company. And this is where they've taken platinum electrodes and slapped them onto a nitinol stent. Again, we're starting to do something different here, but we're still working with the same material technologies. And our last candidate, I very rarely want to say good things about Neuralink, but this is a Neuralink device which for the first time ever started using conductive polymers and new material technologies as their electrodes. Because they had very, very, very tiny electrodes, they wanted very, very many channels. And to do that, what they needed to do was change the way in which we're interacting with the body in terms of the way we're passing charge in and out of it. And this is why. Our biggest limitation we have there is limitation number one. Metals talk in electrons. They pass electrons in electron clouds, but if they reach an aqueous biological system, those electrons stop, and they stop at that surface and they cannot pass directly into the fluid unless you have a chemical reaction happening. And the one thing we don't want to happen is chemical reactions in the body. We can't have those sorts of things happening because then you start to split water, you get the generation of acids, and you get dissolution of the electrode itself. And the more current you push through it and the more that you need to push between that metal and the body, the greater you have the risk of actually creating these reactions. And this is something that hit bionic eyes fairly solidly. That plot up in the top corner there with the hexagonal array with the blue and silver electrodes is a bionic eye electrode array. All the circles there show you the amount of charge it takes to stimulate vision in those patients. And the black line shows you where the chemical safety limit of that material is. So the chemical safety limit is way, way lower than what you need to return vision in the vast majority of patients. So what you do need to do is you need to change the way you're pushing electricity into the body. And the other thing that we have there is that as soon as you put a metal into these soft tissues, whether it's retina, the brain, the cochlea, they're very stiff. And so the body puts up a foreign body reaction. It starts to generate scar tissue. It starts to wrap up that device and try and consume it. It wants to push it out because it's not natural. It's an inorganic material jammed up against a soft, squishy material. And if you have a look at the little brain MRI there that was taken from the Science Museum archives, that's how much a brain moves every time your heart beats. So if you put a stiff metal device, now let's imagine that bed of nail device sitting in that brain, every time it squishes up hard against those really stiff materials. And as that happens, the body generates this scar tissue and says, get rid of it. And that scar tissue is more insulative, so you need more current, you need to push more electricity through it. So you're stuck in this loop of things just aren't working because we have the wrong materials doing the wrong thing. What's really great is that we've learnt to develop these new materials. We now have materials that talk in ions. So conductive polymers, carbon materials like graphene, they're all materials that can interact with ions as well as electrons. And these polymer systems are giving us a lot more opportunities. We're also finding new ways in which they can be manufactured, ways in which they can be scaled up but using existing equipment, because polymers, well, they're all over the world. We have plastics everywhere. And if we learn from the industries that already exist, then we have an opportunity there to really increase and change the way in which we make implants in a really scalable fashion. More than that, we can biofunctionalize polymers. They have chemistries that are quite well easily tailored to different situations. We can put body proteins in them, growth factors, gene transfects. And we can tune their softness. You tune the backbone of a polymer, you can determine whether you want it as soft as the brain or whether you want it, stiff and hard where you need a structural support. They also have image compatibility. So a lot of these organic technologies are able to be imaged in MRI, in X-ray, and they don't have artifacts, so they let us actually understand what's going on with the patient with the device in place, which metals simply don't let us do. And finally, we have drug delivery applications. Now, this can be done with a lot of different types of polymers and a lot of different approaches, and at the forefront, sort of a bit beyond where we're trying to translate now, people are now looking at whether we can incorporate cells, whether we can make devices that effectively grow into each other. Can we take everything we've learnt from like tissue engineering and stem cells and impregnate them into these sorts of devices to create devices that don't just rely on pushing ions in and out, but could actually synapse to the body? Can I put a device on the brain and have it grow down in and create synapses to the tissue I'm targeting? That's our, I guess, the blue sky endeavours, but we're still at the forefront of translation. We're taking baby steps. We're just trying to step away from platinum and into some of these fully functional polymer systems. So I'm just going to now walk you around, I guess, an example of one material that's given us a lot of opportunities just simply in my lab. So conductive polymers are one of these material systems that I've talked about that can pass electricity in the form of ions. These are really great polymers because they don't just pass electricity in ions, they can also be doped to conduct in electrons as well. So you have continuous bipolar ion bands, which means you can put electrons into them from your electronics. It reaches the end of that sort of pathway and hits the body and sees the ionic fluids, and it fluxes with ions instead of building up a double layer and having the electrons sort of stop. So you have these systems now that can mediate that difference between electronics and biology very easily. On their own, they're a little bit stiff. They're a little bit brittle and they fall apart, which is one of the reasons why they haven't been easily translated. What we've been doing is blending them with polyurethanes, specifically with ones that have been medically approved for different devices like breast implants and catheter lines. And in that way, we've been able to produce these, what look like very unexciting blue sheets of material, but they have really high charged injection capacity. They can be used very well for recording. They're also soft and stretchable. What we've done with them is create many different types of devices. We use laser-based fabrication with them because this enables us basically, if you can draw the device, you can make it with a laser. You use layer processing to insulate it and then cut out what you need. And what you can see there across there is basically a peripheral nerve cuff, then an ECOG array. Somebody's playing with one of those cuffs. Most medical devices, you cannot swish and move around like that because they're not very robust. When you make them out of platinum, they're very thin and they're quite brittle and they will break. But when you make them out of polymers, you can take it to a conference, everyone can pass it around, and it will be working afterwards as well. I've done that, it was fun. But you can then start to use other manufacturing processes. Extrusion, that's actually for a pacemaker coil that you've got there that will screw into the wall. The long one with the tape measure is actually a moulded pacemaker wire. And the last one is a wearable electrode, which is made by dip coating. So you start to really look at many different ways and things that metals can't do, you can then do with polymers and provide a lot of different opportunities. Just to show that they do work, we've done implant studies. We put these on sciatic nerve on some rats and showed that over time, what we end up there with comparing them to the platinum, the impedance of the platinum goes up over time as you get that scar tissue layer. And the orange there, the orange squares and bars in that middle graph show us we have a very steady performance over time coming out of those polymer materials. The other thing we can do by making lots of new channels is we can then start to steer the current into the right places where we need it. We have lots of different things we can do then in terms of picking the right fibres to stimulate, and it gives you a lot more flexibility about the way in which you activate tissues. On our ECOG arrays, this is pushing these materials to the limit of how we can fabricate them in size. So these are starting to get down to sort of sub 50 micrometer electrodes, which allows you to pack lots and lots of electrodes into a small space. That's really important when it comes to trying to figure out what's going on in the brain. So these are ECOG arrays designed to detect sort of single cellular structures and figure out where you might be getting aberrant behaviours. So where you might actually have a benefit from having a lot of electrodes that you can't do with a metal technology, to give you a lot more information about what's going on in the brain. And again, what we've shown there is we've done basically at this stage, we're at the preclinical trial stage where we're putting them into rodents and showing that we can detect our field potentials there. That big grid shows you how every electrode on that array is detecting a signal. When it's given a certain input on the animal, it can be recorded on the brain. The next, I think, is a really exciting area that we've been looking at. We've been working with surgeons, and one of the things that's really critical in this industry is not just having guys who, in a materials sense and within a devices sense, can help you make the device and scale it up, but also that you then have an opportunity there to design for a particular patient or a particular tissue. So this is an area where surgeons discovered a while ago that when you amputate, at the end of an amputation, you often get something called phantom limb pain. And when you get phantom limb pain, it's because those nerves that have been cut had nowhere to go. So they kind of roll up in balls and try and find connections and just become this ball of feedback that gives you a lot of pain and you can't figure out what's going on. They figured out that if they took a bit of spare muscle and wrapped it around that nerve, the spare muscle that they were cutting off, because it was going off with the rest of the damage, will grow. The nerve will grow into it and give it actual sites to connect with. And that did is it gave the nerve a nice little spread. You have all these little sites that you can then use. And what we're doing with the surgeons is learning how to put these electrodes in place so we can record what those nerves are intending to do. So if a patient says, I'm trying to create a fist, but they're missing their arm, you can interpret that, feed it back, and so their robotic arm can then make the fist that they want to actually make, right? Then you can also feed back. Through these interfaces and through these types of material technologies, you can then stimulate back up, stimulating the sensory fibres, and let them know when they've got the right pressure, if they've got a different temperature. So this is one where you absolutely need to be working with the surgeons and figuring out how do we make a device that interacts with the tissue but keeps the tissue alive while it's recovering for what is a very traumatic surgery. Long term doesn't cause scar tissue. You want this to be a really functional interface that has to last for a lifetime and operate a robotic limb. So this again, it's a work in progress. We're still in rodents at the moment, but we work with surgeons who do this in human patients all the time. And once we get to the point where we can demonstrate enough of safety and efficacy through a regulatory process pathway, then we are lined up to actually apply these to humans with a device that's been informed by the actual surgery in clinics. The next exciting thing you can do with these exact same materials, I'm just going to check on my time. Okay, not too bad. The next exciting thing you can do is drug delivery, and that's because these are polymers, you can load them up. And we've been loading them up and again working with surgeons to deliver chemotherapy via these materials, and because the drugs are charged, we can push them in and out with electricity and deliver an exact amount of drug right into the centre of a tumour. And where we're finding space in this area is we're working with surgeons who treat glioblastoma, which has about a 14-month survival period. Almost everybody doesn't survive this cancer. And during the biopsy, we put these devices down into the cancer tumours and deliver a known quantity of drugs straight down there into the middle of the tumour, which is something that they've never been able to do before because these drugs are usually delivered systemically to the whole body. They make the whole body very sick, and most of it doesn't make it into the brain because the blood-brain barrier tries to keep drugs out of the brain. But we're going straight through, delivering it straight in there, and we're using a lot less drugs so the patient doesn't get sick. It's an interesting area. Again, these ones have been delivered into rodents, and the surgeons are there waiting for us to get our approvals for our materials because they really, really, really want you to have something to offer these patients. The last area in this that we've been doing is wearables. We spent forever designing these for implants, and everyone kept coming up and saying, but can you just put them on the body? Do they work dry? And it turns out they do work dry, and so we're looking in this space to basically replace those red dot electrodes, which hopefully you've all seen, the squishy ones with the gel that dry out, with just a dry, simple contact there, which is made from foam, a foam of our same conductive elastomer material. And we've shown that we can do the same level of detection, the same level of diagnostics with these dry materials than with them wet. And in this space, we've been working quite a bit with NICUs, with neonatal intensive care, traumatic brain injury diagnosis, as well as it's also been funded through the US DoD over the past to look at things like EEG in people who are in combat situations and understand levels of stress. The very last thing that I want to touch on before I get kicked off the stage, because I'm probably going to run out of time, is where we went with something called living bioelectronics. And that's just taking it that step further. That's saying, can we incorporate cells in these devices? Can we use our knowledge of tissue engineering materials, things like hydrogels, to change the way entirely that we interface with the body? And in this space, we've been working with polyvinyl alcohol gels, with gelatins, which are biological proteins, cross-linking them together and encapsulating stem cells that we then implant at the device interface. What's really cool about this is that normally, and in all these other things that I've described that are implants, from platinum through to what we're doing now with polymers, we basically go in there and we stick a device in an injured or abnormally behaving tissue, which causes it damage, electrocute it, and then expect that tissue to behave in a functional way, which is very, well, optimistic. So now what we're trying to do is, can we grow healthy tissue on the device that we put down in there to help that entire environment recover and be in a regenerative state? Can we do something that actually gets those tissues back to being functional rather than in that diseased, injured state that they started in that required the device? And so to do that, we've been growing different sorts of stem cells in them. We've been tuning the properties by pushing out and figuring out how much biological protein do I need, how much synthetic material do I need to control the mechanics, but also have an environment where these cells can grow. And in this space, we've gotten to the point now, again, we're in vivo trials. We have combinations of glial cells and neural cells growing in these gels that can then grow and create connections with the endogenous brain. And so we've gotten to the point now where we know and can get those connections happening between the device and the brain, but we don't quite yet understand how to control how they happen. And so there's a big area of research we're working in this space. How does this translate? We don't know, and this is kind of where a crowd like you guys are really important in helping us understand, how do we regulate this? There's very clear regulation for devices, and there's very clear regulation for pharmaceuticals and cells. But when you start to put all those things together in one device, there is very unclear regulation, and it's either you have to regulate with every single body to get something to be allowed in a human, or we have to sit down and reimagine what it really means and how we can actually step this into a clinical situation. We also need manufacture and facilities that help us take materials that are non-conformists, that are different, that have biology as well as synthetic materials. How do we create QMS systems for handling those different sides of things and getting them moving together so we can create things that are going to be human-grade implants? This means we've got to be talking to everybody, which is, one of the things I spend a lot of my time doing, which is talking with the clinicians, the patients, the industry, and figuring out how to bring these steps forward so that we can actually utilise what is known, what is really nice technologies that can really improve on our basic devices and get us into a position whereby patients can actually benefit from this. I'm not going to harp on too much about this other than to say there's a lot of opportunity for commercialisation in this space as well. We've very quickly picked up a lot of different areas in research, in industry and in government where we can apply these technologies and are starting to show how, that we can use them. Recently, we've just started a big collaboration with the NHS to get the little caps that are on that. It's actually a fake baby head there in the bottom corner. These are for determining whether babies have seizure when they're born that are in the NICU. The difference between this and a normal device is about 6 hours between the point where a baby is normally suspected of seizure and when it's diagnosed. And that's a crucial window. If it doesn't get the right treatment in 1 hour, it could have lifelong disability. With these sorts of caps, anyone can apply them. You do not need a technical team, a parent, a nurse. Anyone can put them in and get a diagnosis in under 15 minutes. So that's a big difference that it makes in the NHS, and we're very keen to push them forward. And it's simple, it's wearable, so we have a regulatory path for the first time ever that we can actually get these working in humans. So we're really excited about that. Future, we have new material platforms. We need to keep working on this, but we need to get the right people together. Our proper industry partnerships, how we scale up, how we get the right facilities to actually make these work in the clinic. Acknowledgements is I do have a large team of people who actually do this work. It is very rarely me. They get very scared when I step into the lab and wonder what I'm going to break. Hopefully not much. Acknowledgements, a lot of collaborators around the globe as well as here in the UK, and our funders who obviously are the people who make this work. Thank you.